Hello and welcome to the third video of using bioinformatics tools in further education genetics teaching. In this video we're going to start to manipulate our sequences a bit more. So what you should have at the moment is you should have your HOXA1 sequence in full, you should have your HOXA1 exon sequence, you should have your HOXB1 full sequence and your HOXB1 exon sequence. So what we are going to do is we can look at these visually and try to see where the similarities are, but actually it becomes really quite difficult to do so. So what we are going to do is we are going to align these guides together. So we are going to use two different tools to do this. We are going to use this one called Clustal Omega or Clustal O. Clustal O is really good if you want to align more than one sequence with one another. So, as in 4 sequences. You can also look at proteins, DNA or RNA. We're going to be looking at protein. There is also another site called BLAST NCBI. So BLAST stands for Basic Local Alignment Search Tool. BLAST is really good if, for example, you've got a section of a sequence and you want to find it. So BLAST again, you've got BLAST N, and you've also got BLAST X. BLAST X is for proteins, BLAST N is for nucleotides, so we want to click that one. And again, you can search one or you can search two. It's up to you. So let's have a look at Clustal O first. What I really want to show you guys is how you can do the same thing in 2 different places and get very different results. So what we're going to do is we are going to align or compare the full HOXA1 sequence to the exon only sequence. So I'm just going to copy these guys like so. For Clustal and for alignments, I am going to keep the chevrons because unlike my other tools that we have used so far, I've got more than one sequence in my search query box, and my search tool needs to have a way to distinguish between those sequences. So I'm going to use the chevron with my title to do that. So I've got my HOXA1 full, and I've got, the search box is really small, and I've got my HOXA1 exons only. I'm just going to click submit. Sometimes it takes a little while, sometimes it's really quite quick. So let's have a look. You can really clearly see all these little like asterisks here. That is showing you that you've got perfect alignment between these 2 sequences. And where you've got a dash, that means there is no alignment, those nucleotides are missing. So we've got perfect alignment between those 2. That's exactly what we would assume, because in essence, when I've got looked at my exon only sequence on my CDS, I've got rid of all the guff at the beginning and the end. Now I'm going to do exactly the same thing, but this time I'm going to use NCBI BLAST instead. So I've got 2 separate boxes this time. So I'm still going to use the chevron because if you noticed, I want to have a little name on my lines. I want to do the same thing here. So I'm going to copy my full one into my first box. I'm going to copy my exon sequence into my second box. And I'm going to click BLAST. Again, sometimes I've got to wait for a few seconds. And there we go. So this time my summary looks quite a lot different. I have got a little color code here. It's telling me that I've got really high alignment for a section of my query. And then it's shown me the section, but only the section where I've got exactly the same. So unlike in Clustal O, I can't actually see all of my other sequences. I can only see the part of my sequence that is actually in line them. How the same query in 2 completely different tools will show me slightly different results. Another thing that I wanted to show you is what happens when you start aligning more than one sequence. So as we were talking about earlier, like the theory at the moment is that the HOX genes are duplicates of one another. And so HOXA1 is a duplicate or HOXB1 I should say, is a duplicate of HOXA1, so on and so forth, over a very long period of time. So we can have a look at that using multiple sequence alignments. So what we're going to do is we're going to do two things. We are going to first compare our HOXB1 full with our HOXB1 exon, the one that we chose. So we want to make sure first that they are nicely aligned with one another. So I'm going to copy and paste them in there. And I'm going to use Cluster O for this because personally I just like how Cluster O looks. We just need to wait. There we go. And I can see I've got this really nice alignment. However, I start to have not quite perfect alignment in places, and then I've got loads of gaps. And so where I've got like very little alignment. So this is telling me that even though these sequences are meant to be the same, they're not. There's actually big gaps between them. And that's actually one of the shortcomings of a lot of my alignment tools, is that they're not perfect, and it really does depend on your original sequences being good in the first place. So what I'm going to do is I am going to only compare 3 of my sequences. I'm going to compare my HOXB1 full with my HOXA1 exon and my HOXA1 full, because I know that HOXA1 full and HOXA1 exon have really good alignment with each other, as we looked at that earlier. So I'm going to run it, and it's going to run. There we go. So now you can see I've got B1 full at the top, A1 full in the middle, and exon, A1 exon underneath. I've got some areas of like relatively good alignment, starting where the start codons are. That's not surprising. And as you can see here, at this little sequence, I'm just zoom in for you guys. The start sequence here is all, like exactly the same, ATG-GAC. So the first 2 amino acids are exactly the same. After this point, I start to have areas where, there's not so much. But actually, considering I'm looking at 3 sequences, I've got really good areas of perfect alignment. Perfect alignment is where I'm going to see these little asterisks here. So, on the surface of things, yeah, it looks really, really good. Actually, I've got really good alignment between those two. The very last thing that I wanted to show you was how another way in which you can use BLAST. So BLAST is good for if you have a section of a sequence and you want to find the name of it. So let's say that you have a scenario where maybe you just have this section of a sequence. So I haven't got the name, I'm missing the start, and I'm missing the end. I've just got this bit in the middle. And I want to find what that sequence is. I can stick it into NCBI BLAST. I'm going to paste. I'm going to click BLAST. And what is happening now is it's actually searching the whole of the NCBI database for me, trying to find an alignment for my sequence. So it comes out, and what it then starts to do is this time it's going to give me my sequence similarities, and look, lo and behold, HOXA1 is the one at the top. This is really, really clever, and this is the actual one that we used as well. If I go to that one, it's one of the ones that we also used earlier. Essentially, the further down you go, the less likely it is that your answer is correct. Usually you want to look for the one that's fully coverage and it's got really high percentage identity. You can look at graphical summary and it will show you how aligned they all are. And you can then actually look at the alignments and it will show you how it's actually aligned. Like so. So I hope you guys enjoyed this. I hope that you all enjoyed this video and that you have learned how to use some really useful tools. You should at this point know how to put sequences into Cluster Omega, and you can compare those sequences, and you can compare any sequence you want. You don't have to use the ones we are looking at. You can look at any gene and look at these alignments. You know how to use nucleus NCBI BLAST and how to do the same process, and you get slightly different results. You can also use NCBI BLAST to look at and find sequences when you don't have the entirety of that sequence as well. I will see you all for the very last video that is going to be on searching for protein structures. Have a nice day.